Okay, so you want to be a brain surgeon. You want to be a brain scientist. You want to be a neuroscientist. Um, and you want to explore the brain, cut open the brain, take apart the brain. So, um, one place to start is learning how we study the brain. Okay, and um, even before this slide, really, the only way to study your brain, and somewhere in your chapter they talk about a man named Phineas Gage, and well worth looking up on the internet if you want to know Phineas Gage's story. I'll give you the brief story, the short version. Um, Late 1800s, railroad worker, good worker, good family man, worked on was laying track, and they were working with some explosives, and a rod with a point on the end, uh, called a tamping iron, basically. Um, and I think it was like 6 feet long. It During the explosion, it went flying through the air, and it went in Phineas Gage's cheek, kind of behind his eye socket, and out his. I think it was his right frontal lobe, and he lived. It didn't kill him. So uh, he, you know, got up. He was bloody, all that kind of stuff. They took care of him. He went home. Uh, Eventually, came back to work, and his personality changed. So, uh, as I said, a hardworking family man. Now he was prone to vulgar language. Was not a good worker, and uh, left his family, and just had a lot of problems. He just wasn't really a likable person anymore. So, what conclusions could you draw from that? Well, uh, something in his motivations, his morality, uh, his personality must have been in that frontal lobe area that was destroyed after or as a result of the explosion. So he ended up, he ended up being I think he was like a circus act. Come see the man who had a hole in his head. And but really, I tell that story because that's really the only way people were able to study the brain uh, for a while. Like you damaged that part of the brain, and this happened. You damaged this part of the brain, and that happened. And then kind of use the same technique, but on animals. That's getting to this slide: a brain lesion, destroying brain tissue to study animal behaviors after such destruction. So basically, you take part of the brain out, and so what happens to that animal's behavior? Okay, so it sounds like fun, right? How long could you go about doing that? Just cutting up animal brains and seeing what it does to them. Uh, then we look at clinical observations. Okay, clinical observations on these are done lots of ways. So maybe after autopsies, okay, you look at patients who suffered from certain psychiatric diseases, neurological diseases, and you looked at their brains after they had died and make comparisons between healthy brains and the not so healthy brains, and looking at those differences. So this image is quite the storehouse of brain tissue. All right. So then we get a little bit more advanced here with an electroencephalograph that's always abbreviated EEG. And this is looking at electrical waves as they go across the brain surface. Okay? So it's measuring, we talked about that electrical activity that's going on during an action potential. All right, so that's really what's happening. It's identifying kind of where in the brain there is more electrical activity, and at any given time the amount of electrical activity kind of moving across the brain. we've gotten pretty specific as we've put electrodes on very specific areas of the brain now, even inserting electrodes inside the brain. This picture is, the electrodes on the outside of the brain, but inserting electrodes on the inside of the brain, figure out exactly when a very specific part of the brain is activated. Okay, I gotta pause here for a second. I'll be right back. Okay, where was I? EEG. Okay, so electrical activity. So one thing about EEG, it doesn't allow us to look really inside the brain. It can tell us things about brain activity. Now it can tell us about maybe where activity is located, but fairly primitive compared to some other things that we have going on now. So a PET scan, positron emission tomography scan. So what happens here is that during you get injected with some radioactive blood sugar, basically, and then when you are doing certain tasks during the scan, you can see where the radioactive blood is going in the brain, and then based on the level of glucose in a particular part of the brain, you see where that activity is. Okay, I should have a picture of that. I guess I don't. All right, so the third way is the magnetic resonance imaging, and this can tell us a great deal more. You can see the details that are located on these slides here. So what this is showing is the center of a schizophrenic patient. Okay, so you can see the ventricle that is on a normal patient compared to that of a schizophrenic patient. This is a common occurrence in the brain of schizophrenic patients. Okay, and this one is showing what happens when a patient lies. And there are companies now that actually are saying that they have lie detector machines different from the ones that have been historically used that get to 80, 90, even seen over 90% accuracy on whether or not someone is lying. So that's you can look up lie detector machines and learn all about those. But it's based on some of this MRI scan technology that suggests that when we lie and we know we're lying, that information comes from a different part of our brain, and then when we seek out a piece of information that comes from the truth. Okay, I think that's it. Yep.